Welcome. This UPathDB tutorial will explain how to define results based on their genomic location in relation to another set of results. All UPathDB databases are built with the same web architecture, so they look similar to each other, and functions, such as the one described here, are the same across all databases. For this tutorial, we will use plasmodb.org. Using the feature described here, you can visualize different data types in relation to one another. For example, you may be interested in locating SNPs relative to a particular set of genes, or DNA motifs, like restriction enzyme sites or transcription factor binding sites, based on their location in the genome relative to genes, or transcripts that are antisense relative to annotated genes. For this example, we will search for EcoR1 restriction enzyme sites located in the 500 base pair upstream region of protein-coding genes in P. falciparum. This search requires a three-step strategy. Step 1 is to perform a gene search looking for protein-coding genes using the gene type search option. In Step 2, we will find all EcoR1 sites using the DNA Motif search option. Step 3 is to define genes from Step 1 based on their genomic location in relation to the genomic location of the results from Step 2. In this example, genes with EcoR1 sites upstream of their start codons will be defined. Let's begin with a search for protein-coding genes in P. falciparum. Under Identify Genes By, expand Gene Attributes and click on Gene Type. From the page Identify Genes Based on Gene Type, choose Protein Coding and limit the organism to P. falciparum. Naming the search helps you keep track of each step while you are building a strategy. When you are done, click Get Answer. The query results for Step 1 include 5,418 protein-coding genes. Let's move on to Step 2. To search for EcoR1 sites, click Add Step. From the Add Step pop-up window, choose Genomic Segments and then DNA Motif Pattern. Enter the DNA motif GAATTC, which is the recognition site for the EcoR1 restriction enzyme. More complicated motifs can be input using Perl-style regular expressions, which are explained at the bottom of this Add Step pop-up and at the following link. Next, choose the organisms that you would like to include in your query. This sets the parameters for the DNA motif search. Now we have to choose how to combine the searches. Combine genes from Step 1 with the genomic segments from Step 2 by clicking on the option labeled 1 relative to 2. After clicking on Continue, you are presented with a pop-up window that allows you to configure the relationship between the two search steps you just ran. At the top of the pop-up, you will notice a logic statement that needs to be carefully considered. There are 5 user-defined parameters in the logic statement: the data type to be returned from the query, the location of the returned data type relative to Step 2 results, the relationship between the data types from Step 1 and Step 2, the relative location of the second data type, and the strand location of the second data type. Each parameter has several settings to choose from. You can choose to return items from Step 1 or Step 2 of your strategy. When you make a selection, the changes are shown graphically in the box below. Step 1 is represented in blue and Step 2 is in red. Choosing Genomic Segment from Step 2 in the first option box initiates changes in the graphic box and rearranges the order of search results in the logic statement. When we change the option to return genes from Step 1 instead of genomic segments from Step 2, corresponding changes are made in the graphics below. You can define the relative location to be used in the search. You can search the exact region of the gene, or you can search upstream of the gene, or downstream of the gene. Custom search areas can be defined relative to the start and end of your gene from Step 1. For example, you could search the entire gene as well as 1,000 base pairs past the stop site, or the upstream region plus the first 500 base pairs of the gene. The relationship between the data types of search 1 and 2 is chosen here. The two segments can either overlap. The first search result can contain the second result, or the first search result can be contained within the second search result. The same choices, exact region, upstream, downstream, and custom, define the second data type region parameter. The orientation of the second data type can be set to either strand, the same strand, or opposite strands. Getting back to our EcoR1 example, we want to arrange the logic statement to return each gene from step 1 whose upstream region contains the exact segment from step 2 and is located on either strand. Click Submit once you have arranged the logic statement. The first search for protein coding genes returned 5,418 genes. The second search, the motif search for EcoR1 sites, returned 7,354 segments. Combining these searches based on their genomic locations returned 214 genes whose upstream regions contain EcoR1 restriction enzyme sites. Gene IDs that meet the parameters of your search strategy are presented in the bottom table. Several columns in the results table contain information regarding the relative genomic location of the search results. Match count is the number of times the motif matched sequence in the search region. Region gives the genomic coordinates and orientation of the regions searched, and matched regions gives the exact coordinates of the matched motifs, in our case the exact coordinates of the EcoR1 sites. Thank you for watching this UPath DB tutorial. Please contact us with any questions or suggestions.